organisms, also create products that are not drugs, ditto for pharma. So basically, biotech companies are usually small and new, but not always. This shows revenue in billions. I went over before. Note that there is some overlap. So Amgen, which is we'll visit this in the spring semester for our biomanufacturing class, with a revenue of 129 billion, is very close to AbbVie, the classic pharma company. Every one of these pharma companies has a biotech division. How does this start? We talked about this last time. Generally, somebody gets an idea, and then you make a pitch. So there's all kinds of venues where investors are sitting in the audience, and you get up there with your little slide deck, and you say, here's this cool idea I have, and here's the data we have. Here's our IP. IP is intellectual property. These are things like patents, things that you own. So you show all of this, and hopefully somebody out there says, well, this sounds really cool, and they set up a meeting with you, and if you're lucky, they give you $100K or something to help get the product going. Obviously, as this thing gains steam, angel donors are usually get people off the ground. This is something very early on. So angel investment, obviously, very, very high risk, but massive turnout. So if they get it right at an early stage, they can own a significant percentage of your tiny little company. And then when you grow into the tens of billions, these guys now make big. So about 10 years or so, you get a private market. Usually startups are bought out by bigger established companies. This is very, very common. So once the product you have or your idea looks sufficiently attractive and it's passed a number of regulatory hurdles, and it looks like it's going to make it, then pharma will actually start to compete, and they'll make bids on your company. Okay, they'll either buy you out, period, and this is where all of whoever's involved. So if you guys are all employees in some small company, and you all have stock, and the stock was maybe 5 cents a share, and somebody buys you out at $5 a share, well, you guys all go shooting up 1,000, 100. So bringing drugs gross to market value of the company. Initial public offering is called IPO. So this is when the company says we're now going to sell portions of our company as stock to investors. So you go on the open market, and people start, like retirement funds, et cetera, start buying up your stock. Price goes up. Jobs in biotech, research and development. This is where I work. R&D is basically very, very scientific. We're going to stay close to the literature, come up with ideas of what potential drug intervention for different diseases. Preclinical, everybody understand this. Clinical means that they're going into humans. Human clinical trials. Preclinical means before humans. Obviously, the entry, if you have some drug that you design, before you can put it in people, this is extremely rigidly controlled. So there's regulatory agencies, the FDA, Food and Drug Administration, that reviews all your data and says, yes, you can go into humans, or no, you can't. And if they say no, they usually tell you a whole bunch of steps that you need to do in order to proceed. Okay, I forgot it. So preclinical, this is usually cell-based studies, animal studies. So lots of little rodents get whacked here in disease type models, which we'll talk about later. Discovery research, bioinformatics plays a huge role. Bioinformatics, if you guys will remember, is genome. So it's looking at genome sequencing information. What do the 22,000 odd genes for humans mean? What do they do? Do they have different functions at different stages in a human's life, etc.? That's all bioinformatics. Operations, process development, manufacturing, production. Well, the Amgen facility is a production facility. So they basically are operations. There's no research goes on there. It's all scale up and process development and production. Quality, this is a huge topic that comes up in the spring semester. Quality control, this is called QC, and quality assurance is QA, QA/QC. This is all involved in every company is required by law to police themselves and to ensure that the product they are producing is the same, meets stringent requirements every single time. And there's knockout criteria, so if something goes wrong in the production stream, remember there's many, many, many steps, so problems can go wrong just about anywhere, and the quality of your particular drug is compromised, then you have to have a process for rejection, okay? And this is all part of QA/QC. Clinical research and regulatory affairs. This is now where you're going into humans, and you're starting to look at clinical data, and this is going, usually, you start off in, what do you call it, normal people that do not have disease. Then later clinical trials is patients with finance, administration, obviously legal, the list goes on and on. So biotech and pharma today are gigantic entities. Jobs, basically education level, and plays a pivotal role. So analysis, this is like QA/QC quality, production, working huge machinery, QA/QC, R&D. R&D is research and development. This is very early. So this gives the different diplomas. Obviously, in R&D, this tends to be much more cerebral, and so students that enter in at a much higher level of education. So usually a Bachelor of Arts or Science or a graduate degree is much more common in R&D than in the other specialties. Okay. And salary is usually tracks along with it. This is just giving a typical organizational structure of a medium-sized biotech company. The R&D, operations, quality. This is QA/QC again. Clinical research, finance, administration, legal, facilities, etc. Typical type of scheme. Pharmaceutical companies and biotech companies can be massive, with dozens of buildings scattered across often international global sites. Salaries shown for typical PhD. I think this data is a little bit dated. I think this is a little on the low side.
They all seem low.
Yeah, this looks low. I'll have to check. Anyway, I got this off of some website. So 56K, masters about 41, PhD about 43, and that's across the nation. Some areas will be quite a bit higher than that. So what's the best approach to landing a good job? Professor Bosey goes over this more in the 101 resume writing class. A degree in molecular biology, biotech, or informatics. Minor, you should have some sort of informatics base to your skills. Through skills, oral and written communication, obviously, because you have to survive an interview, and then you have to survive if you're called back to the company if they like you, and they want to quiz you again. And the people that will be interviewing you usually go right up the ladder, so you're going to have brilliant people there that know way more than you do, especially about what they are working on, and they're going to be probing you to see, can this person think on their feet? Is this person expressive? Do they make sense? Or are they a space shot? You know, trying to figure out if you want to hire this person. Collaborative team player. All, especially industry, biotech, as well as pharma, extremely team-oriented. Regularly, you'll sit down at the table with people that are in modeling, chemistry, biochem, and you'll have no idea what they're talking about. It's a completely different science, a different language. The whole point is we used to have something at the company I worked called lockdown, where the whole team, which is all these people with different expertise, were put in a room and said, you guys figure out, solve this problem and figure out what you're going to do. And nobody leaves until you finish. And these could go for 12 hours. It's not uncommon. They bring food in. You're allowed to go to the bathroom. That was called lockdown. So being a team player is key. How do you interact with people and how do you guys talk and discuss, come to an agreement, make a plan. Biotech workforce is attracting so many people because of good pay, reward, and in general, expanding industry. Current global crisis, this is one of the questions on the quiz, which I went over many times. Expanding global human populations is huge, right? Remember, we are currently consuming 125% of the world's resources. So the total of what the world can produce, we're consuming 125%. Obviously, this is unsustainable, okay? Furthermore, the population continues to grow roughly exponentially. So the 125% gets larger and larger and larger with each passing year. So all kinds of effects, extinction, toxicity, pollutants, overfishing, practices that are unsustainable. This cannot continue. We need to do something. Well, one thing we can do is genetically modify organisms. This was also and the idea is, can we, for a given acre of land, produce more? Because we have more people. Can we get bigger ears of corn and bigger crops, more stable, that don't spoil as fast, global foreign policy, more energy, and all these things obviously playing a role. Depleting fossil fuel reserves such as petroleum, water availability, freshwater supply is critical, and many nations on Earth, and finally human. So all these things are problems that biotechnology attempts to address. Estimate about 10 billion people by 2050. Obviously, something can occur before then. This assumes that we keep the same trajectory we have now. Current global human population, it's up to 7.9, I believe. I'll show you that. It's on food law. Food law, I'll show you. U.S. today is about 350 million people. This is an interesting statistic. We have just under 5% of the world's population, but we consume over a quarter of the resources. So every U.S. citizen is consuming way more than the average world citizen, right? That's because we have a great country, lots of GDP. I want to show food law so you guys can see it. The website is given there. There's food law right here. Here's food law. I'm sorry, 7.69. So it's similar to the number I just showed you. So food law site, the link is given, and it gives you human population. So roughly, 2 or 3 babies are being born every second somewhere in the world. This is total net growth. So people are dying at a slower rate than people are being born, and that's why this number keeps going up. So we're around 7.7 billion right now, and we're going to keep going up unless something catastrophic happens. Now, there's tons of other stuff in this site. I'm not going to go through it all. It has deaths and accidents and environment and garbage dumps and all kinds of things. That's pretty wild. 7.7 million. This is a quote from, I've heard Albert Cash. He's my counterpart in Kingston. He always gives this quote. It's one of my favorite. The man who has bread has many problems. The man who has no bread has one. The reality of starvation is it trumps everything. British Petroleum Annual Report on Oil Reserves estimates about 53 years. This number is highly variable. It's been estimated at about 50-some years for some decades. And obviously, we keep going. So more and more reserves are being found, but obviously eventually this is going to be Donald Kennedy, former director for American Academy of Master Science. A solution to sustainable source of energy may be the most significant challenge for future human survival. Because obviously what we're doing now cannot continue. Today's politicians, this is a key topic in this course. Establish processes and policies tomorrow. So the guys down in DC are basically making decisions that are scientifically based. And the question I think all of us have is, how capable are politicians scientifically? Are they literate scientifically or not? And the answer, as you might guess, is highly variable. Let me give you some terrifying examples. Some of you may have heard this from last semester. So there's a wide disparity between knowledge of general public about DNA and biotech, the actual science and applications. What kinds of things do politicians say? These are all quotes taken from speeches. Here's Tom Aiken in 2012 states that women cannot get, this is barely scientific, but this is common knowledge. Women cannot get pregnant from ready, quote. It seems to be, first of all, from what I understand, that the doctors, it's really rare. If it's legitimate rape, the female body is raised trying to shut both of them. It's a complete crock. All you need to do is look at the facts. Approximately 32,000 rape pregnancies in the U.S. each year. Sarah Palin, another one of my favorite for these inane quotes. Sometimes these dollars go to projects, she's talking about R&D expenditure, and NIH, National Institute of Health, et cetera, that are government funded. Projects that have little or nothing to do with the public good. Things like fruit fly research in Paris, France, I kid you not. Moron, you cannot say this. This is scientific, total scientific illiteracy. Fruit fly research, most of what we know about the genome, the genes that have been identified came from fruit flies. Just because it sounds like it's a trivial organism, remember, bear in mind that fruit flies are somewhere around 80% identical DNA sequence to us. So we're not that different. So fruit flies have most of the same genes we have. Sequences may be a little bit different just because we diverged so long ago. But you cannot make this statement. This basically says that she has no understanding of so-called basic research. So basic academic research often studies things like mold, Neurospora crassa, fruit flies, E. coli, etc., and to reject it all as being irrelevant is absurd. Basically rejecting the large bulk of genetic research that's available to them. Laura Bacher, is there some thought being given to subsidizing the clearing of rainforests in order for some countries to eliminate that production of greenhouse gases? I can't even believe that you could make such a statement. It's the exact opposite. I think what he's confusing, you may have heard, that some respiration, does everybody understand this? We were talking about this in biology class last week. So remember, you start off things like CO2, water, and from this you can make glucose. For example, I think Dr. Bose wrote the formula out for you guys last week. This is a glucose. Could be galactose. There's a number of C6-containing sugars, but glucose is the most popular. This process, which is described in biology, is energetically hugely uphill. Remember, that's a key concept of bio. Run in this direction, shown in black. This is called photosynthesis. The opposite direction is what we do. We humans, we run this line this way. So we eat food, and we blow off, we exhale CO2 and water. These are so-called end products of primary metabolism. This process is called respiration. Now the problem with respiration, which all of us do, is that we exhale a huge amount of CO2. Burning of fossil fuels, for example, taking gasoline, which is similar to glucose, very, very high up on the chemical potential energy scale. And when you burn it in your car, you're producing CO2 and water, and a lot of other stuff which make up smog, like pollution. So it is true, plants do a certain amount of respiration, but by and large, a huge amount of what they do is photosynthesis. So plants mainly run in that direction. So if you clear rainforests, you're reducing your ability to consume CO2, not make it. You understand? So this statement is absurd. He's got the thing absolutely backwards. Even though maybe 1% of what plants do is respire their own sugar sources, 99% of what they do is fix. Got it? This statement is ridiculous. Scientists all over the world say the idea of human-induced global climate change is one of the greatest hoaxes perpetrated out of the scientific community. It is a hoax. There is no consensus. Wrong. 99% point to all the things that are happening. They say climate change is a reality, and it's within our generations are the ones that are going to either deal with it or not. All that stuff I was talking about, evolution and embryology and the Big Bang, all lies straight from the pit of hell. Really? What science says is the temperatures peaked out in 1998, and we've gone for over 10 years with the temperatures going down. No. Totally fallacious. Poor damn clown. Mars gets more things wrong than Mars is essentially the same orbit as Earth. Mars is somewhat the same distance. We have seen pictures from our canals. We believe in water. If there's water, it means there is oxygen. If oxygen, it means we can breathe. These guys are, this is the VP, former vice president. These are the things they say. So the blunder they're making, and again, Professor Posey is teaching you guys this, is H2O. This is water, right? Water, it is true, it contains oxygen in it, but anybody knows who sticks their head underwater and tries to breathe, the presence of O2, diatomic gaseous oxygen, these are two totally different things. Just because oxygen is there does not mean you can't breathe, right? It is time for the human race to enter the solar system. Don't move on Earth. I love California. I practically grew up in Phoenix. The future will be much better off tomorrow. Okay, government policy, funding. NIH, I should have spread, is National Institute of Health. They fund most of the scientific basic academic research that is conducted, as well as some work that's done ultimately, for example, in small businesses. All depend on public opinion and politicians around the world who are either approving or not supporting this type of thing. So scientific method, and this was established a couple centuries ago. This is the scientific model. Again, we went over this one. Observation. Hypothesis, so look at what's going on, establish laws or patterns. Say, okay, I think these laws fit the following type of structure. That's a hypothesis. Make a prediction based on this hypothesis, design some experiment, and then see whether or not your hypothesis is correct. Now, as everybody knows, hypotheses that continually are proven correct time and time again for a long period of time become theories. And eventually, after huge periods of time, they can become laws. So most of what we have in science are theories. These are explanations of how it works. And obviously, think revision can occur if an experiment gives a result that the hypothesis does not predict. So you can either alter it or you can reject it, find another one instead. This is the so-called scientific. And this is, again, a picture we put over in classes, observation, hypothesis, experiment. Experiments always consist of a control group in which no manipulation has occurred, an experimental group where you're changing something. Results, they either support or they're inconsistent, and then eventually this becomes scientific theory. So almost 3,000, this is 5,000 years ago, Parmenides, formal description of nature, Socrates, applications of philosophy to nature, politics, including knowledge. And this is a great Alhazen, about 1000 AD, father of modern scientific method. So about 1,000 years ago is when this approach first appeared. Experimental data, reproducible results, make conclusions, and see how the world is working. All this stuff should be uploaded, by the way. It should all be on Sakai, and I put chapter 2 up there as well. I just put it up about half past 8. Copernicus, heliocentric solar system, meaning the sun is at the center, 1500. Kepler describes planetary motion. Galileo, father of modern science, is keen observer, who, for example, saw moons orbiting Jupiter, etc., and made a number of observations that were inconsistent with the Holy Roman Catholic Church at the time, and hence he was basically imprisoned, house imprisonment for end of life. 1700s, new Leibniz, this was development of new physics, calculus, theory of gravity. 1800s, Lamarck, Lyell, Darwin, Faraday, Dalton. So this is all as things became more and more established. Organisms change with time. They're not static, violent. Geological forces have shaped and are shaping the Earth today. This is part of tectonic plate movement, which obviously is a major force today in shaping the world. Origin of Species, published in 1859, fundamentally altered biology for all time. Darwin bet, well, we'll talk a little bit about that. Faraday, combustion and chemical reactions. Combustion is basically taking sugar or wood or petroleum and converting it to CO2 and water. Tremendous amount of heat. It goes boom, depending upon what you've got on the right-hand side of the picture. Dalton with atomic levels. Okay, 1900s, Einstein relative to space. This is the space-time continuum. Haber-Bosch reaction is one of the most important reactions which led to basically supported huge human population expansion. This is the conversion of nitrogen. This is atmospheric nitrogen. Remember, about 80% of the air you breathe in is N2. N2, very stable, almost totally unreactive, very difficult to do anything with it. He came up with a process for converting it into fertilizer. This is reduced nitrogen. Instead of N2, it's either oxidized, nitrates, nitrites, or ammonia, which is reduced form. And the Haber-Bosch reaction basically enabled us to suddenly get more and more productivity out of cropland. Right? It was ammonium nitrate, etc., could be used to increase crop yield. Alexander Fleming discovered penicillin. Watson and Crick in the 1950s. Thomas for successful bone marrow transplant. John Gurdon, one of my favorite, very colorful scientists we'll talk about later when we get to the nuclear transfer chapter. Thomas Alva Edison. I have not failed. This is when he was said, Dr. Edison, don't you find it horrifying that you spent so much time and you failed so much trying to invent a light bulb? I have not failed. I have just found 10,000 ways that don't work for his invention of light bulb. Finally, of course, discipline, innovation, persistence, intelligence, curiosity, devise theories in science. You try never to guess. Obviously, imagination is part of innovation. Enormously important. It's your ability to see things in a way that nobody has seen them before, or to look at some fact in your textbook or something that you just happen to think about. You see something on TV or on the internet, and you say, that's odd. Dr. Chris said X, Y, Z. I wonder if I can exploit this in some manner. So imagination, innovation, creativity, very important processes towards getting this thing going. But to move and advance science requires a lot of persistence and hard work. Theory, analyze different set of facts, identify general principles, scientifically acceptable principles offered. Beliefs, these are not the same things. Mental acts, condition of habit or place of trust in a person or thing, faith. So beliefs are things like religion, for example. Theories, Big Bang, gravity, general relativity, evolution, these are all theories that have been basically proven, more or less proven time and time again for decades, and belief systems, one example. So what is life? Any definition tends to be biased. The exceptions, again, we went over this in biology class, lecture last week. Biological information system capable of replication with variations. Variation implies natural selection, ability to adapt. If you're producing the exact same individual's offspring all the time, you're not doing the population any good. Because the key to understanding Darwinism is that the population is not static, it's a variable. And all the individuals that make up a population are slightly different. And some of them will have benefit at a given time and others will not. And that causes a flow where the genetic structure of the population changes with time. And this is essential to all life. Carbon-based, water-based, it's everything on planet Earth. Cells, consumes and produces energy, and adapts. Again, scientists discover liquid oceans of water on Mars. What is life? So Darwin, Charles Darwin, 1831, set out, he was the son of a preacher, set out to find evidence for the Great Flood. He made observations which did not support physical creation. I'll go over this later on in the semester. Many observations, for example, in South America, that were completely inconsistent with immutable species with no variance. So Darwin's arguments for divergence, exponential population increase, or superfecundity. Almost every species that have been examined have the ability to grow exponentially. So something must occur that limits their continued expansion. Predation, disease, death, something. perceived stability of populations, limited resources in space, so all these things are creating basically a structured types of populations, struggle for existence. This is the so-called natural selection process. Uniqueness of individuals, individual variations inherited and passed on. So each one of us are producing genes that we have within ourselves, and that collection of genes, this is called germline or haploid cells, so eggs in female humans and sperm in male humans, basically has the individual's genetic makeup in it. And everybody here is going to have a slightly different collection of either eggs or sperm that they produce. And that is defining what the future generations will look like. Differential, survival, or selection. Not everybody thrives and not everybody breeds. Over countless generations, the population changes. It's not the same. Part of Darwin, in each generation, populations produce more offspring than there are adults. Populations do not continue to grow in size. Food and other resources are limiting. Individuals within populations vary. Many variations are hereditable. Over time, population changes as events, and this is all part of adaptation. Evolution of life, biological patterns, change is consistent. Over 99% of all species that have ever lived on Earth are dead. There have been 5 mass extinctions over the past few 100 million years, and we're currently in one right now. This is called the Anthropocene, or the human-induced mass extinction. So humans have killed off already almost a quarter of species on planet Earth. You do not believe in evolution, you either understand it or you don't. So I've had students say, Dr. Chris, do you really believe in evolution? No, I don't believe in it. You either look at the facts and understand them or you don't. It's not a belief system. DNA manipulation exploited by humans. This is the basis of molecular biology and biotech. The origin of life on Earth. Somewhere around 2,500, 2.5 or more billion years ago. This was, the spontaneous generation from Greek. 1920s, which again, we go into this more biology. Simple carbon-based compounds such as CO2 by the light. DNA is hereditable material. Some of this we're going to overview in the next couple chapters. So this is Stanley Miller, who did the bottom-up, and those of you remember the bio lecture. And Yuri Oparin is not mentioned, Oparin as well. Simple primordial earth compounds from amino acids. The famous Hurst-Chase experiment, DNA proved to be genetic material. Watson encrypted DNA forms a double helix, proposed mechanism of DNA replication, which has withstood the test of time to this day. Crick basically established central dogma of molecular bio, and that is DNA. That's the RNA. Protein. That's all life that we know. Some viruses go backwards for a step or two, but basically all life has to do this. This is the dogma of life. DNA contains information, forms RNA. This is, you can see, think of it as a more mobile form of the exact same sequence, and this forms proteins. And proteins basically are proteins and enzymes in you dictate your structure and your metabolism. That was Crick, this is the RNA world, which we'll talk about shortly. Carl Versa, we went over this also in biogenetic code. RNA is primordial genetic material. What does this mean? Most scientists believe that the first life on this planet had RNA as the genetic material, not DNA. It's simpler, and RNA actually has catalytic function. So RNA is believed to be how life started. We don't know because we can't see it, but we can hypothesis a bunch of experiments support it. Alton and Czech basically identified ribozymes. Ribozyme is RNA. Catalyzes reactions. It's was not understood or accepted actually until almost a decade after his time. It took a while for people to believe. Wally Gilbert, great Wally Gilbert, Nobel laureate, coined the term RNA world, which means life started off doing the last two steps of the 2016, 355 genes are identified defining all life on Earth, derived from the LUCA last universal common ancestor. So the belief today, this is part of this is a top-down coming from metagenomic analysis of sequences of life all across the Earth, is that LUCA started off with this number of genes. All of us have in it. Again, this is basically starts at humans appearing, moves back in time into the Mesozoic, Paleozoic, and finally Precambrian. Remember the Precambrian explosion is where all the major phyla or major groups of life appeared. This is about 500 million years ago. Okay. And this leads to the tree of life. The LUCA, about 3.7 billion years ago, was somewhere down here. And then all of life as we've experienced it on Earth is shown by this tree. Again, this is Carl Sagan. I went over this bio where each month is about 1.1 or 1.2 billion years old. So what Sagan did is condense, because it's so difficult for us to comprehend this enormous expanses of time. As he said, let's take 13 point some billion years, which is the age of the universe, and condense it into 1 year. What does it look like? So each month, everybody understand? So each month is about 1.1 or 2 billion years. So planets are appearing somewhere here in August, and then by the 9th month, first single-celled organisms are appearing. LUCA, multicellular organisms. These are simple things like sponges and small multicellular creatures. They are appearing in November, and then December is where everything basically happens as far as life goes. The Cambrian or Precambrian explosion occurs on the 15th. We go through all these different periods. Apes appear. First human ancestors walk upright at about 9:24 p.m. Homo erectus appears. Anatomically modern humans appear 6 minutes before midnight. Homo sapiens have lasted for 6 minutes on the cosmological scale when condensed into it. Implications of human genome, medicine, pharmacy, anthropology, psychology, total Neanderthal DNA sequence known from this girl that was approximately dated about 18,000. Psychology, psychiatry, forensics, insurance, Angelina Jolie, double mastectomy, based on having a BRCA1 allele that put her at a very high risk for breast cancer. DNA sequences, 1995, Haemophilus influenza. This, by the way, is a prokaryote. Remember, these are bacterial cells. Before nucleus, this is a much simpler cell structure than is present in us, for example. Cenorhabditis elegans, this is called C. elegans. This is a round worm, about 19,000 genes were sequenced. Drosophila, 19,000 sequences to death. Homo sapiens, about 25,000. This number has come down, and it's now about 22K today. Mouse, 85% identical, sequence both DNA and protein to human, 2002. And rice, about 44,000 genes. Question.
How are they so over for the Homo sapiens?
Pardon?
How are they so over for the Homo sapiens?
They're actually way. So before, because I'll try to make this as simple as I can. So you can do something called two-dimensional gel. You look at isoelectric focusing and staining and a molecular weight, and you form a slab. And you can take some cell that you guys have, or organ tissue like biopsy, munge it up, run it out on the gel, and you see tens of thousands of spots, which are non-identical. So from those types of studies, originally we thought that there was somewhere around 100 to 100 50,000 genes in a human. This is what we believe based on those type of experiments before the Human Genome Project. So we thought they were a huge number because the genes are different. These all look like different proteins. However, it turns out, this did not come out until the Human Genome Project, that one gene can go to form many proteins, especially in eukaryotes like us. This is called splicing. We'll talk about it a lot later. So we can make many, many different proteins from the same set of genes, just by shuffling around what parts are being expressed. There's a calcium channel, there's a calcium channel gene, which has 41 different splice exons. And so all dozens of different calcium channels can be made from the same gene. That's why. It's taken a while to determine the actual number of true genes. And remember, this is not easy to do. 3 billion base pairs in the human genome, and only a few million of them are coding for sequence. H1N1, this is influenza virus. About 50 to 100 million humans died. This was in 1918. Killed far more people than World War I did. Equivalent of this, if this happened today, if about 4 or 5% of the human world population died, this would be about 300 million dead. Potential for global devastation. This experiment horrifyingly was done. You can actually engineer influenza today. It's a great story, but I'll go into it when we get to biowarfare. Talk about biowarfare, I hope later this semester, if not spring, they don't have that. But you can take existing avian flu, change 5 genes, and this could kill up to 50% of the world population. So just by making 5 changes. The scientists that tried to publish this was refused to publish because of the tremendous potential for biowarfare. Bioinformatics, data mining, these are all ways of examining huge masses of data or sequence. Signal transduction, I'm sorry, I have an extra word. Signal transduction basically means cells are responding to signals from the outside. This could be as simple as nerve impulses, like so if you step on a tack or you hit something hot in the oven and your foot jerks up. That's signal transduction. It's the neural, interneuron transmission that is telling your foot to pop up. Pharmacogenomics, this is basically how each individual human, based on the sequence you have, is responding to meds. Not the same. Xenotransplant means putting something into you that did not come from a human. So pig valves, hearts, etc. from other organisms. Combinatorial chemistry, a lot of these are beyond the scope of this class. Proteomics, same as genomics, except looking at protein sequences. Biochips, I presented this a little at last lecture. It's this thing about so big and you have a little chip, which can have hundreds of thousands of spots, and it allows you to determine things like DNA sequence, SNPs. SNPs are single nucleotide polymorphisms that are the difference between all humans. 0.1% is about 3 million base pairs. Tissue engineering, the agriculture, biotech, all different kinds of things that can be done. So this is part of ethics, genetic editing of humans to produce super athletes. This is illegal, and this is basically accepted around the world. It's an incident in China. It's called the task. Cloning, theoretically, we make dollar. You could make a human. This is difficult to do. A primate was just completed, I think, about a year ago. So the first primate was cloned. So theoretically, you can take DNA out of a human and create a whole new human with the identical sequence. Who should decision makers be? Scientists, politicians, God forbid, their knowledge of these things is atrocious, as all of us hope for them. Administrators will move. Who decides whether these things can be done or not? Oh, I'm going a little bit over time. Why don't we all take a break? And we'll reconvene after. Is Larry Summers, former president of Harvard University. I believe when history of this period is written, 250 years from now, what happens in life sciences and biotech during the next 25 years or so is likely to be a large part of it. And obviously, this has already been proven to occur. The remaining slides we went over already. This is your assignment, which hopefully nobody found too difficult. And I already showed that video in bio. Whose model we have? Okay, chapter 2. Any questions about chapter 1? Genes and genomes. Okay, so this is all part of molecular biology. Molecular biology is usually called mole biology. Molecular biology is basically the study and manipulation of nucleic acid, so DNA and RNA. A lot of DNA manipulation. Okay, so what I have to do in this chapter is basically bring everybody up to speed as to what DNA is. And I'll give as little detail as I can, but hopefully enough so that this stuff is comprehensive. So DNA encodes all of the genes that provide instructions to control activity of all cells. Genes influence behavior, determine physical experience. Go over cells. Molecule of life, specifically nucleic acid. Chromosome structure. RNA protein synthesis. This is part of CRIC's DNA world. Right? RNA world, DNA RNA protein. Mutations causes the consequences. Something called the epigenome, which has been under very intense study for the last 20 years. So first, cells. And again, this is a crash course in cell biology. Single human has somewhere a variable number around 70 or so trillion cells. Approximately 200 different cell types. Largest human cells are neurons. Approximately 3 feet running from your spine to your toes, depending upon how tall you are. That's can be a single axon, a single neuron can be this long. All human cells within a single human have the same sequence, unless you're popping off cancer cells, in which case sequence is variable. Different cell types express different genes, typically about 5,000 genes at any given time. So for example, if I collect all of your red blood, all of your white cells, say neutrophils, particular type of leukocyte, a white blood cell, and I look at the genes that it's expressing, it'll express only about 5,000. And the genes expressed by a neutrophil will not be the same as a muscle cell, a neuron in a brain, etc. Many genes will overlap, so-called housekeeping genes. But many genes will be unique to that particular cell, and a particular time that left the function. So obviously, a leukocyte that's responding to a bacterial attack is going to be different from a quiescent leukocyte that's just roaming around the circulation, not doing anything. So this is variable. Okay? Now, all cell types will be a little bit different. All cells have plasma membrane, cytoplasm, and organelles. Organelles are occurring in eukaryotic or so-called higher order cells like us, not in prokaryotic. Prokaryotic cells like no. Plasma membrane, double layer of lipids that surrounds the outer surface. When we're talking about protosomes in the first life on Earth in bio, some sort of liposome or lipid sphere with an inside and an outside spontaneously formed. We believe that's very led the way. Cytoplasm, inner contents of cell between nucleus and plasma membrane. nucleus obviously only present in a eukaryotic cell, organelles, structures that perform some kind of function. Life on Earth is distinguished between bacterial cells or pre-nucleus, i.e. no nucleus present, or true nucleus. Two great types of cells. Viruses are not included. Some scientists don't classify viruses as being alive because they actually don't do their own metabolism. They rely on some host cell to do it for them. So prokaryotic cells, bacteria, archaebacteria, size is generally quite small. No nucleus, DNA is located in the cytoplasm, no organelles. Higher order cells, eukaryotes, protists. These are protists are by and large, not totally, but some, generally small single-cell structures such as plankton in the sea. Fungi, plant, animals. Larger by a good 10-fold in diameter, and because it's acute, the volume of a cell is about 1,000-fold larger. Eukaryotic cell much larger than bacteria. DNA enclosed membrane around the cytoplasm called nucleus, many organelles. Typical bacterial cell where there's some nucleoid region, not membrane bound. And there's ribosomes. Pili are these little protrusions coming from the cell. There's a plasma membrane. This is if you cut the surface, there's an outer cell wall, plasma membrane, and then the inside. So this is classic cell structure. Inside, outside, and they're separated. They're separated by a lipid envelope which restricts movement. This defines inside, which is alive, and then outside, which is not. Many bacteria are flagellated, and in mycoplasm, you guys can go over flagellated. Eukaryotic cell showing an animal on the right, plant. Immediately, it should look much different to you. Large vacuole. This is a nucleus. The dark mass in the center of the nucleus is called the nucleolus. This is where RNA is made, ribosomal RNA, in huge amounts, because all cells have to produce lots and lots of ribosomes, or else they cannot do the last step of CRIC, DNA RNA protein. This is catalyzed by ribosome. Ribosomes are made up of rRNA, that's the ribosomal RNA, plus a bunch of proteins. These are massive machines. That's the nucleolus. So animal cell has all these things. Mitochondria actually look, here's a mito here, they look like bacteria. The theory is that these in fact were bacteria a billion years ago. Do not memorize all these, but you should know the basic differences between these two cells. Plasma membranes, fluid dynamic complex, double-layer barrier, include cell adhesion, communication, shape transfer. Basically establishes the inside of the cell, which is alive, and the outside, which is the surroundings. Okay, so it's a permeable, somewhat permeable, but selective barrier. Certain kinds of things cross easily, other things do not cross at all. Cytosol, nutrient-rich gel-like fluid, which is present in all our cells, and organelles, compartments where chemical reactions, cell processes occur. And in eukaryotic cells, these are highly organized, so you can think of it as different buildings in a production facility, where there's going to be a power plant, and then buildings that do different kinds of things. These are organelles within the cell. Each organelle has its own biochemical focus. A quick overview of plasma membrane, cytoplasm, ribosomes, rough endoplasmic reticulum, and the kinds of functions that they have. And everyone should know these. You have a rough view. Smooth ER, especially bioclass, we spend a lot more time going over this. Golgi apparatus, the rough, smooth, the Golgi is basically a series of stacked membranes that are involved in protein production, maturation, and transport. And again, I can't go over this class. I assume you guys are roughly all this. Lysosomes contain hydrolases, or just enzymes. Peroxisomes are basically for oxidizing. Microtubules, these are highways along which organelle structures are transported. For example, liposomes, so small lipid particles containing synthesized protein. They're transported along these microtubule highways across the cell. So there's a superstructure and an organization and flow of materials constantly in the cell. And it's occurring on the millisecond to the second. Microfilaments, intermediate filaments, two different types of fibers, and centrioles, opposite paired with chromosomes, center of chromosomes. Cilia, flagella are involved in propelling cells. Humans are obviously flagellated sperm, which swim. Nucleus, nuclear envelope, nucleoli, chromatin, central vacuole in plants, chloroplasts also present in plants. Give plants their green color. Okay, so quick overall review of what we just went over. Two main different types of cells that comprise life on Earth. Prokaryotes, no nucleus. This is pre-life, so it's believed to have come first. A billion years or so go by, and then you have a eukaryotic cell, much larger, highly organized, packed with organelles and structure, much different from a bacterial cell. Okay? Now this does not mean higher order. It just means it appeared later in the evolutionary scale. The bacteria basically dominate this planet Earth, more so even in eukaryotes. That's a quick overview. Get a nice basic feel for these subcellular structures and what they do. It'll come up again. So 1869, Meischer coined the term that nuclein could not be broken down by proteases. To include nuclein, it's not a protein. Nuclein has acidic properties. Acidic properties means releases a proton, right? Proton concentration goes up, pH goes down, and becomes acidic. Negative charge at neutral pH means a proton got lost. Definition of acid. So they were renamed nucleic acids, term we use today. Two types of nucleic acids identified very early on. DNA, RNA. Very, very similar. Two little differences. Go over that. Everybody got it? Everybody know what is a protease? Everybody knows what it is. Anytime you see, these are enzymes, and the ase at the end means that it hydrolyzes. So water is added across, and you turn many beads on a string into separate monomeric units. Proteases break down protein. So nuclein and nucleic acids are not chopped up by proteases. Proteases, we have massive amounts of them in our gut, stomach, pepsin. Pepsin was a protease. And every time you eat a hamburger or steak, the proteases, the pH goes down because proton concentration goes up. And the proteases are cleaving these huge proteins that make up beef, muscle, whatever else is in the horrible stuff you guys eat. Breaking it up into little amino acids so that you can use the amino acids to make your own protein. Metabolism and other functions. Proteases do not cleave nucleic acids, DNA or RNA. The simple experiment said, no, it's not protein, there's something else that contains key information within a cell. Griffith, the famous Griffith experiment, 1928. I'll go slowly just to be sure everybody understands. He took two strains of bacteria using both Streptococcus pneumoniae, which is the causative agent of pneumonia. So this is a pathogen. And in microclass, a bunch of students, a bunch of you guys will actually culture this organism from your mouth. So many of us have this pathogen in us, but it's not doing much. That's why we're healthy. He had one strain caused virulent pneumonia, causing smooth strain cells, and harmless cells that were rough. This is how the cell appeared. So smooth actually was a capsule and pathogenic. Rough cells are not. So we took these two strains, everybody understand? These are small variations of the same species, but they're different a little bit. And that difference gives you killing cells versus harmless cells. And we looked at these two and he did the following experiment. S cells are capsulated, that makes them smooth. R cells have no capsule. Did an experiment in which mice, I have a slide coming up next, which I hope will organize this for you. Mice were injected with S strain, death, pneumonia. R strain, mouse is fine, non-pathogenic. This means the mouse's immune system cannot beat this guy, but can beat that guy. Same with us, when we get a sufficient amount of S pneumonia in us, we either live or get real sick very fast. He heat killed S strain, so all he did boil them. So they're dead. And mixed killed S strain with live R strain and asked the question, do the mice survive? Got it? We heat kill the lethal organism and we mix it with the one that's non-lethal. What happens? Everybody understand? So I ask it on every quiz, or maybe I'll say it to you. One never knows. So here it is. Live S, inject this guy, rest in peace. Live R, fine. Immune system successfully takes care of R. Heat killed S, guy's fine. All right, so if you take the lethal strain and boil it, this guy survives. Understood? You need a live lethal strain in order to kill the mice. So once you boil it and you kill the cells, they're no longer infectious, incapable of replication. Agree? Some people are shaking their head. We take dead S cells, which you already know don't do anything, right? This guy's fine. We mix them with live R cells. What happens? Rest in peace. From the dead mice, you haul blood out and look at it, filled with S cells. Do you understand? This is a critical experiment. And it indicates two things. Number one, whatever happens upon boiling is killing the cell, but not some critical information component, answer nucleic acid. The nucleic acid is not destroyed by boiling. Proteins generally are. Nucleic acid is not. So that survives. That's number one. Number two, genetic transfer. Somehow, the rough cells are drawing DNA out of the dead cells and are becoming smooth and virulent. DNA exchange, DNA transfer has occurred. This key experiment basically established the foundations of biotech, because this says you can transfer DNA between organisms. In this case, it's different strains of the same species. But this experiment provides not only the recognition that something that's heat stable, nucleic acid, is got genetic information in it, but number two, DNA transfer can occur between organisms, even bacteria. Every time. Two key experiments. One.
So if the heat-killed S cells were put into mice that have no R cells in them, nothing can happen.
Right, that's this experiment. Number three.
Oh.
If you heat kill S, they're fine. Excellent question, because obviously, if you had not done this experiment, this is called a control, you wouldn't know that killing them, boiling them, makes them biologically non-infective.
Right.
So the only conclusion from experiments 3 and 4 is that something is transferred from the dead S cell to the live R cell, producing S strain type. That means DNA transfer can occur between organisms, and this experiment was done in the 1920s. That was the dawn of molecular biology. So transformation, DNA is taken up by somebody else. Heat treatment broke open as and the capsule, DNA is released, DNA is heat stable. It was not destroyed by boiling. This has been known for a long time. Nucleic acids are stable to boiling, proteins are not. Living R cells took up sDNA, transformed the properties of R cells so that they became virulent, and achieved the capability to make capsules, which renders them resistant to the immune system. Foundation of biotech. This mimics transformation process to introduce proteins of human therapeutic interest into bacteria by transferring DNA. We can take genes of our cells, for example, insulin. We can stick it into a bacteria because the genetic code is universal across all life. That bacteria will now chug, 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 and make insulin. Human insulin, even though it is single cell, it has no interest in insulin. Everybody understands insulin is a hormone in us that regulates blood glucose levels. So the bacteria is not gaining any benefit from this gene. But because of Griffith's experiment, he paved the way, saying this kind of thing can be done. It took a couple more decades before we humans actually did it. Avery with Claude and McCarty, basically evidence that DNA is inherited genetic material. Homogenized matrix bacterial cells, treated extracts with proteases, remember these, ACEs are hydrolyzing big long biopolymers into individual building blocks. So little nucleotides, single nucleotides, or single amino acids. Proteases, amino acids, RNases digest RNA, and DNases digest DNA. We have all these enzymes in us because the food you eat, for example, if you're eating plants or meat, you eat material that has protein, DNA, RNA, lipids, and carbohydrates, all the constructions of a cell in the food you eat, with the ability to degrade everything. In translation experiment with extracts, found that extracts from heat-killed S treated with DNase mixed with living R do not transform. So once you chop up the heated DNA, it now is no longer capable of transforming R's into S's. So this again, 1944, this is about almost 20 years later. And further established that DNA is a genetic material that's being transferred between viruses. The experiment provided additional evidence that DNA material improved the transforming factor in the Griffith experiments. Bacteria. DNA structure. Building block of DNA is the nucleotide. So again, think beads on a string. All DNA is a long, long biopolymer. This can be hundreds of thousands to even millions of nucleotides linked together, beads on a chain for millions long. That makes up our chromosomes. Specific sequence that is replicated every time. Each nucleotide is composed of a pentose. Pent means five-carbon sugar. This is called deoxyribose. It also has a phosphate, and it has a nitrogenous base present. Nitrogenous bases are interchangeable component of a nucleotide. Each nucleotide contains one base. A, T, G, C. 4 letters in the DNA alphabet. Universal across all life on this planet. Okay, we get into chemical structure, which you probably don't know much about. So what that is. Here are 5 carbons. This is called deoxyribose. Carbon 1, 2, 3, 4, 5. The 5 C's linked together by bonds. These are called non-polar covalent bonds, which Dr. Bosey will talk about sometime this semester. So 5 carbons make up the pentatose. This one is called ribose, specifically deoxyribose. This is the same sugar, but called ribose, present in RNA only. And if you look, the only difference between these two, ignore the phosphate for the time being, is that at the 2' carbon, deoxyribose has no oxygen, whereas ribose does. There's an OH here, and in this position there's an H, no oxygen, hence the name 2-deoxyribose. And that became DNA, 2-deoxyribose nucleic acid. And this is the number one fundamental difference between RNA and DNA, is the presence or absence of that hydroxyl group. Got it? I realize you don't know much about structure, but these are rings. It's a cyclic version of the sugar. Nitrogenous base, 4 types, A, T, G, C in DNA. For RNA, very similar, one tiny little difference. So purines are adenine and guanine. So adenine and guanine are both bicyclic. Try and get the basics. Bicyclic, and pyrimidines are cytosine and thymine. RNA does not have thymine. It has instead uracil. That's the second difference between RNA and DNA. Whether or not there's an oxygen or a hydroxyl group in position 2', and whether there's U or T. Both of these form the exact same hydrogen bond pair, uracil and T, both hydrogen bond to adenine, and cytosine hydrogen bonds to guanine. So the base pairing is exactly the same, but the one, U and T are swapped. And if you look, you'll see, even if you don't know anything about chemical structures at all, you can see that these two are very, very, very similar. The only difference is there's a methyl group here on T, and it's not present on T. Only difference. Forms the exact same hydrogen bonding structure, pairing the two bases together. Okay, so 2' OH versus not present, and U versus T. Okay, Bruce Watson and Crick, with it back when they published this paper, both received a Nobel Prize about 9 years later. This is the actual model that they built. It shows the stacked bases that are rotating around in a double anti-parallel double helix, which is their gift to mankind. It's the structure of this. Student testing. So how did DNA structure occur? Shard gap. A whole bunch of people were involved in generating the individual pieces of data, which enabled Watson and Crick to propose this model. Number one, Shard said the number of G's in any sample of DNA and the number of C's are exactly the same. And the number of A's and the number of T's are exactly the same. But these are different. They're not always the same. They're often different. But B matches this and this matches this. Got it? So if you take any stretch of DNA and look at it, you'll see maybe 1,000 of these. That means there'll be 1,000 of these. You'll see 650 of those. It means there'll be 650. Franklin and Rosalind Franklin, was a woman, obviously, Maurice Wilkins, provided arbitrary crystallographic data that showed a helical structure and also showed key atomic dimensions. There were 3 of them. And using those dimensions, Watson and Crick came up with the anti-parallel double helix. Watson and Crick used data from Franklin and Wilkins, Shard gap base pairing rules, and developed a model that revealed the definitive structure. This is way before molecular modeling. Today, you can do all this with molecular modeling. So computers, you can form these types of structures. Molecular structure of nucleic acids is structure for deoxyribose nucleic acid. It's the title of their paper published in Nature Scientific Journal on April 25th, 1950. Nucleotides are joined together to form long strands of DNA, beads on a string. Two strands joined together, wrap around each other, form a double helix. Nucleotides and strand are held together by phosphodiester bonds. More on this in just a second, I'll explain it. Phosphodiester bonds connect deoxyribose sugar molecules to the 3' and 5' end. Each strand has a polarity. In other words, if a DNA strand runs in this direction, it has a distinct polarity. So it has a 5' end and a 3' end, and they are distinct. So if you take any strand of DNA, it will have a beginning and an end. And in DNA, they run in opposite directions. So one strand runs this way, the paired strand runs the other way. So it's called anti-parallel. 3' and 5' I'll explain in a second. Each strand has a polarity. Polarity refers to the carbons in the sugar molecule and which direction it is running. And it's coming up in another slide. Two strands of DNA molecules are held together by hydrogen bonds. This pairs A with T, or U, and G with C. Adenine pairs with T, guanine pairs with C. That is why the number of A's always equals the number of T's, because they're matched. So however many of those you have, the other strand will have a T there, so it'll be exactly the same. However many G's will match C. And these will not be identical. There'll be some random mix. Two strands run in opposite directions. The polarity is reversed. One runs 5' 3' that way, and the sister strand runs 5' 3' the other direction. DNA resembles a twisted ladder, which is called a double helix. Okay. All came from watching in Craig's Nation. Okay, let me explain. This people, I think, had some starters. So each one of these is showing a single sugar. You can tell immediately this is DNA as opposed to RNA because of two features. To be sure the class is still conscious and able to respond. What are they? So how can you tell if this is DNA and not RNA? First, everybody look at it for a while and think. Come up with an answer independently. Molecular biologists will look at that instantly and say it is DNA and not RNA. Way in the back.
There's a lack of uracil. There's no uracil.
Correct. You have a T here, and a T here, and no U. That's number one. What's number two?
Looking at the left rung, or the left side at the top, we have a 5', and at the bottom we have a 3', and then on the opposing end, you have 3' and 5'.
Sorry, let me just emphasize. Remember 5 carbons in ribose. 1, 2, each vertex is a carbon atom. And Dr. Bosen teaches this spring. You have to. 1, 2, 3, 4, 5 is 5 carbons. Continue.
The 5' at the top, and then at the bottom we have a 3' end, and on the opposing side we have the 3' end at the top, and then the 5'. So that is the anti-parallel characteristic of
Total
DNA.
agreement. So this basically shows that one strand runs this way and the other strand runs that way. But it turns out RNA does something very similar. So that statement does not distinguish between the two. What you said is true, but it's true for both RNA and DNA. RNA generally is single-stranded, but has double-stranded structure, which is critical to the RNA world. So the statement you made, though true, does not distinguish the two. As a student in the back said, we have T's and no U's.
I don't understand the 5', 3'.
I'll talk about that later.
Okay.
Anybody else get Hakeem?
No, I don't.
Perfect. C1, C2, C3, C4, C5. C2, no hydroxyl, deoxy. DNA is 2' deoxyribonucleic acid. No OH, otherwise you'd be an OH here. If this was RNA, then an OH. No OH, and T's not used. Everybody got it? Okay, polarity. What Watson and Crick observed and proposed is if you look, remember, there's a sugar-phosphate backbone, and it runs continuously and makes huge, long pieces of DNA, which can be hundreds of millions of nucleotides all in a sequence. This makes up your chromosomes. Hundreds of millions. Sugar, phosphate, sugar, phosphate, sugar, phosphate, millions times. These are called phosphodiester's. And if you look at the ribose, remember 1, 2, 3. At the 3' position is a phosphate. And at the 5' position is a phosphate. Polarity. You can immediately see that for every nucleotide, there's a phosphate on a 5' end and a 3' end. So there's a direction. Got it? The direction, which when we talk about DNA synthesis, always runs 5' to 3'. This is called in frame, or the correct reading frame and direction. Just like when you read a book, you go from left to right, same with DNA and RNA. You'll always go from 5' to 3'. More on that in a second. So that's what polarity is. If you look at this end, it has a 5'. If you look at the other end, it has 3'. DNA grows in this direction 100% of the time, no exceptions across all life on Earth. Always grows 3'. That's the 5'. So you add on to this end. Okay, did that answer the question? So polarity is not the same. You can look at this end and say it's different from this end. It grows in this direction. Any questions? Everybody have it? Please study this.
Dr. Chris.
And make sure you have a fairly good feeling for directionality, deoxy, 2'-deoxy, and U versus T.
The explanation that I gave, it included not only the polarity, but I also mentioned the anti-parallel and the fact that it's a double helix, whereas
Correct.
RNA is just a single helix.
A complete agreement. But RNA forms hairpins and loops and extremely complicated structures, and it has the exact same anti-parallel structure.
Right.
It can. So generally, RNA does not form anti-parallel different strands, but it can fold back on itself. It does this all the time. So the statement you made is correct, but it's not distinguishing. Now, it is true, DNA is essentially 100% of the time, unless it's being replicated, it's called the replication bobble or fork. Unless it's being replicated, it's always double helical, whereas RNA is not. It will have sections that are single-stranded, but it will have big sections that are double-stranded. And that's the basis of three-dimensional structure of RNA, and that basically allows Shannon Altman to hypothesize the RNA world. So what you're saying becomes critical very shortly.
Okay, so
Especially in bio.
when it caps off, that's when the RNA can become double-stranded.
Correct.
Okay,
If you
and
take
it
an
closes.
RNA strand which goes 5', 3',
Yeah.
and it forms a hairpin,
Mhm.
now this strand and this strand are running in opposite directions, and they make a perfect
Understood.
double helix, just for short. It's called a stem loop structure.
Yep.
That's critical to the RNA structure. Why the whole RNA will. So this is showing the double helix again. RNA will do this for short sections. Everybody got it?
Yep.
Everybody, is this all right? And note, G and C always paired, and A and T. Tough question. Why are G and A always portrayed as being longer, and A, T, and C are shorter? Why?
Because angier variants and larger.
Fused bicyclic structures, so they just take up more space. Everybody understand? So if I go back, I don't have to go back at all, you can look up here. This is G, and this is called a purine. It's a fused bicyclic structure. This is wider. So that's why this is being depicted as a wider molecule. G is wider. The pyrimidines are monocyclic, and hence very physically shorter. So when you look at these stacked bases, the purines are taking up more room. That also says why G and an A cannot pair. So if they pair, you'll cause a bubble. you'll put a bubble in the middle of the helix. And that's extremely denatured, non-stable, and DNA polymerase will repair it. Okay? Why don't we pause here again, and we'll take a break until 11 o'clock. That gives you 10 minutes. Can you please? Okay. So DNA is double helical, and you can think of it as one strand running in this direction, and one strand is running in this direction. Right, anti-parallel. These are connected by hydrogen bonds, and hydrogen bonds are linking A with T and G with C. As Watson and Crick stated in their Nature paper, this means that it's fairly easy to break this. Hydrogen bonds are not covalent, they're much weaker. So it's not that difficult to break these two apart so that they're single-stranded. And we do this all the time. We can do this, for example, by heat. So in a PCR, polymerase chain reaction, you rely on the fact that these things will come apart and become single-stranded pieces at elevation. Run in opposite directions. Think of it as beads on a string. So at each one of these positions, there's an A, a T, a G, or a C. And these are connected by phosphodiester bonds. These can be hydrolyzed. I'll talk about it in a second. The addition of water, this is done, for example, by a nuclease, will break these apart. Whenever you eat food, like a salad or banana or something, you're hydrolyzing the DNA that makes up the cells and the stuff that you're eating. Okay? The phosphodiester bonds, obviously on the other strand, will be the complementary pair. So A will have T, this will have an A over here, this will have a C, and this will have a G. And on the other strand, right? Those A's and T's are always paired, and G's and C's are always paired. Okay? Everything got that? Then, last item is ribose. Ribose is a 5-carbon sugar. This is 2,5-deoxyribose. And again, Professor Posey will go over a lot of this in the spring. This is called C1, 2, 3, 4, 5, and it's usually called prime. Prime means that you're counting within the ribose, or in this case, 2,5-deoxyribose, okay? The beads on a string are caused by phosphate esters. This, by the way, is usually simplified with just vertices. So this means the same as that. Again, 1, 5, 2, 5, 3, 5, 4, 5, 5 deoxyribose, because there's no OH. The N is part of the nucleic acid, and this is either a bicyclic purine or a monocyclic purine. Okay? Either one of them, only two choices. One is A or G, and the other is C or C. These hydroxyls are not the same. One comes off the 5' carbon, and the other comes off the 3' carbon. This gives DNA directionality. These H's become phosphodiesters. So this forms a phosphate group. And the next bead on the string will be the next deoxyribose. Take N with some base over it. This is called the phosphodiester. It's this structure. Phosphate and these two oxygens have an alcohol from a carbon. This is called a diester. Now, most of the reactions that we do, for example, during digestion, is hydrolysis. So by simply adding water, a molecule of water across this bond, you can generate a free alcohol or a free phosphate. So add addition of water. Most of the reactions that occur in the human body are simple addition and subtraction of water. If you do that in a coordinated manner, you get life. Okay, so this is the diester form. If you hydrolyze it, you now are breaking those bonds. That's what nucleases do, and that's was part of the Avery experiment that you saw. They boiled S organism and then treated with DNAs, and you would instantly lost the ability to make smooth particles, smooth capsule. Okay, that's just a rough picture of what we're doing. It has a direction, and they're not the same, and they're anti-polar. Any questions on this? Anybody? So gene is a sequence of nucleotides that provide cells instructions to synthesize either a protein or some type of RNA. There are many, many types of RNAs. There's messenger RNA, which makes protein. There's rRNA, tRNA, microRNAs. The list goes on and on and on and on. And more RNA types are being discovered all the time. So there's many types of RNA. On average, genes are a few thousand nucleotides long. And since it's a triplet codon, that means that most amino acids are on the order of a few hundred amino acids. Make up most protein. Genes influence how cells, tissues, organs appear and defines the term trait. So a gene gives you a trait. For example, blue eyes, brown eyes. Height is usually multiple traits. So there's bunches of traits you're inheriting from your mother and father that make you either tall or make you short or something like that. Not all genes are used to produce protein. Some genes make ribosomal RNA. This is called transfer RNA, and this is called microRNA. There are many of them. There's silencing RNA we'll talk about later. Chromosome structure. Chromosomes, where cells package DNA, and this is occurring in the nucleus of eukaryotic cells. Chromatin, strings of DNA and DNA binding proteins called histones. So in eukaryotic cells, we do not have the naked DNA alone. It is wrapped around a protein core. Histones are highly positive or basic, and so a lot of the interaction between wrapped DNA and histones is electrostatic, plus and minus coming together. State of DNA inside nucleus when cell is not dividing is in this chromatin form. During cell division, chromatin is coiled into fibers that wrap around each other, so chromosomes are highly coiled. Condensed chromosomes are more easily segregated in daughter cells during division. So the total length of DNA in a cell is huge. It's many times bigger than the cell itself. But by supercoiling it and wrapping it around proteins and forming these complexes, you can make DNA chromosomes very short. Okay? These are ways of condensing. Bacteria do something similar. They also supercoil. So it's like taking a rubber band and twisting it, and you end up with something very short and snarled. And that's how DNA is most of the time, especially during division. Because you can't have something that's miles long. You need something that can be packaged in a very small space. Most of our cells have two sets or pairs. So most of our cells have a total of 46 chromosomes, or a pair of 23. One comes from your mother and one from your father. The number of chromosomes does not change with generation. So all humans have 23 pairs or 46 chromosomes. In germline, this is ovaries for females and testes for males, the number of chromosomes is halved. This is called meiosis, or reductive division, which we'll talk about in body. So germline, this is cut in half. This means every sperm or egg that you are producing will have a collection. Some of the 23 will be from your mother, and some will be from your father. Now, there's usually a few mutations per generation, so you will have some sequence that's absolutely unique to you that is not present in either your mother or your father. These are mutations, all part of that. These are called diploid or two-end homologous pairs because, remember, a mommy and daddy are humans, and they are 99.9% identical sequence. That means that your home chromosomes coming from mother and father are roughly 99.9% identical. If your parents happen to be siblings, this is illegal, this is called incest. If they are siblings, then instead of 99.9, it will be a bigger number because mother and father are the same. One reason incest is not allowed is you're not getting mixed in DNA, and that's considered same blood, illegal in most countries. Autosomes, chromosomes 1 to 22, and sex chromosome is the last pair, X and Y, right? Each female has two X's, one inherited from one of your two from mother, and the other X comes from your father. And if you have a Y, then you're a male, and you will have a Y and an X. Gametes or sex cells containing a single set of 23 or haploid number, productive division. All other cells in the body are somatic or diploid cells, and examples are kidney, neurons, muscle, etc. Characteristics common to eukaryotic chromosomes. So again, we have 23 pairs. Chromosomes consist of two thin rod-like structures called sister chromatids. Exact replica of each other copied during DNA replication. DNA synthase, which removes water and makes phosphodiester bonds, is an incredibly accurate enzyme and makes very few mistakes, maybe 1 in every 3 billion. So there may be one mistake out of an entire human genome during replication. Exact replicas during cell division, each sister chromosome is separated. Each chromosome has a single centromere or region consisting of intertwined DNA and protein that joins chromatids together. Centromere delineates two arms, usually called a P and Q arm. Each arm of chromosome ends with a telomere, which is the end. We have linear chromosomes. Bacteria do not. Bacterial chromosomes are circles. We have long lines. Well, there's an end. The end is called the telomere. Highly conserved repetitive sequence. Telomeres allow cells to divide without losing genes during age and cancer prevention. Telomeres become shorter. So my telomeres are shorter than yours. I'm older than you. As more cells go through more generations, they become shortened, and as they get really shortened, you end up with deficit. This is called old age. Things start going wrong because you're losing genes during the replicating process. Typical chromosome, this is replicated. Here's the centromere, and this shows replicated arms. This is done under replication. P and Q, replicated chromosome, and if you, in the nucleus for humans, there will be 23 pairs. Potential consequences of shortening of telomeres during the aging process of DNA gets lost or become damaged, and this leads to deficits, things going wrong. Okay, this is non-replicated. So this shows you a single piece of DNA. There's a centromere portion somewhere in the middle. And this is also chromosome number 1. One is coming from your mother, and one is coming from your father. Okay, and these are randomly mixed. So although there's one mother and one father for every one of these pairs, remember meiosis is separating us in half. We don't know which one came from which parent unless you do forensics, sequencing. Then you can say, This is the paternal chromosome, and this is the maternal. By looking at sequence and comparing it to the families. Prokaryotype analysis is studying chromosome number and basic aspects of structure. Spread them out on a microscope, you get a picture like this, and you can number them all. They were numbered in terms of decreasing size. The biggest is number 1, and the smallest is number 22. X and Y are unique, right? This is the sex chromosome. X chromosome is huge, and the Y chromosome codes for basically enzymes involved in making testosterone, androgens, and little else. So chromosome size, highly variable. 1 and 2 are the largest. Male Y is the smallest, and obviously 20, 21, and 22 very small too. Is this particular cell haploid or diploid, and is it somatic or germline? This particular cell, assuming that this is a karyotype for one cell. What you've done is smeared, and you've stained it, and you see something that looks like this after you organize everything. So is this haploid or diploid, somatic or germline? What does haploid and diploid mean again? What's a haploid cell? And what's a diploid cell?
Diploid is the full amount of DNA for the human, and haploid is half the.
Correct. So is this haploid or diploid? How many chromosomes are up there? I mean, if you bother to count them all, if there's 46, it's diploid, and if there's only 23, it's haploid. This is diploid. This is diploid. Because every one has a paired partner. So one of these is your mother, and one of these is your father. It's confusing because after DNA replication, when the cell's starting to divide, these will form an X shape, hence the name chromosome. And that means it's been replicating. It's still diploid at that point, but there's two that are almost the same, but they're 0.1% different. One came from your mother, one came from your father. Unless it's consanguinal, then your parents are siblings, in which case it'll be much higher than 0.1% different. Got it? 46, this is a diploid cell. In the germline, eggs and sperm for humans, there will only be one of these present, one of each. Sometimes paternal, sometimes maternal, making up a single head of a sperm or a single unfertilized egg. It will be a collection of one from your mother, others will have one from your father, etc. And then some of your eggs will have an X, and others of your eggs will have another X. No Y, or you'd be a male, right? Sperm, on the other hand, roughly half of them will have an X, inherited from your mother, mother of men. Y is inherited from your father. Got it? Half the sperm will have that, and half the sperm will have this. Okay. Somatic or germline? This is the same question reworded to see if you guys know the nomenclature. Somatic means this is a cell in your body. The answer is yes, because it has 23 pairs and 46 chromosomes. Germline would have to be haploid, where you had only one of each of these pair, and you would have either an X or a Y, or if it's an oocyte, female egg, you have two different X's, one inherited from your father, one from your mother. Got it? Keep cooking. Okay, here's a typical karyotype. And you look at it and say, oh my God, look at that. I've got 3 chromosome 21s. This is called trisomy 21. It's very common, relatively common. And this is called Down's syndrome, occasionally called mongolism, but that term is no longer in use. And it leads to a typical Down syndrome patient, which I'm sure everybody has seen. So occasionally, anomalies can occur during the meiotic process where a mistake happens and one of the chromosomes gets mispaired. So you end up with 2 21s in either an egg or a sperm. So when they fuse, you end up with an incorrect number. It's called trisomy 21. Trisomie is usually seen in smaller chromosomes because if it occurs up here, it's usually embryonic lethal. So you cannot form a living organism. The 21 is one of the few that survives. There's a few others. For example, sex chromosome. You can have XXY or XYY. DNA replication, somatic cells divide by mitosis, which is a cell division. One cell divides into two daughter cells. Each one is identical to the parent in sequence. And they occasionally do a single error to the DNA codons. Single human skin cell can produce many cells with 46 chromosomes per cell by continually dividing. That's how we got to be as big as we are from single cells and tiny little embryos. Cell division, which is an exact copy, gives you a human with 30 to 70 trillion cells. In order to complement this DNA, the parent cell must be copied by a process called mitosis. DNA replication, sex cells divide by meiosis, creates 4 daughter cells that will be sperm or eggs. These are called gametes, and these will be haploid. DNA in each daughter cell is not identical to the parent because it's halved. So some will have more father genes, some will have mothers. There'll be a random mix. Some will have X's and some Y's if you're a male. Chromosome number is cut in half. Each daughter cell has only 23 chromosomes, no pairs. Pairs have been separated in meiosis. Through sexual reproduction, fertilized egg forms between male and female, sperm and egg by mitosis, and you end up with the full complement again. Half came from the father and half from the mother. Okay, this was first proposed by Watson and Crick. Semi-conservative replication. This turns out to be the mechanism that it is. You end up with parent DNA. ATs, double helix, anti-parallel, running in opposite directions. Remember, 5' 3, you can't tell from this picture, but they're running in opposite direction. Usually by convention, 5' is on the left-hand side. 5' 3, 5'. Something happens, and the DNA denatures or comes apart. This is catalyzed by our cells that are divided. These come apart. You then fill in monomeric beads. Link these together. This is a condensation or dehydration reaction which forms phosphate diesters between. That gives you the sugar phosphate bond. DNA polymerase runs off millions of these every time one of our cells divides. One cell is adding on this way, and the other cell will add on in the other direction. At the end of DNA synthesis, you end up with 2 exact copies of what you started with. One strand came from one half of the DNA ladder, and the other came from the other half. And the part in the middle has been newly synthesized. You've now exactly replicated the chromosome. Now the cell can split, and the 2 daughter cells will be identical. Only during meiosis are they non-identical. They should be identical. And this shows that DNA string are coming apart, and then one of these is growing 5' to 3' this way, and the other grows 5' 3' the opposite way. You have to accept this right now. The DNA synthesis always occurs with the 5' end onto the free 3' OH, so it always grows towards the 3' end. That means that when you open the DNA up, it's synthesizing in this direction on one strand, opposite direction away from the fork, replication fork, where this is opening up. Got it? So steps in DNA replication. Unwind the DNA. We do not heat up our cells to 95 degrees. We'd all be dead. But an enzyme called helicase breaks the bonds and essentially opens up and unzips DNA. Single-stranded DNA binding proteins is called SSBP, for single-stranded DNA binding proteins. Stabilize the single strand so it can't come back together. This gives DNA synthesis a chance to make separation of strands occurs in regions called origin of replication. Adding short sequences of RNA are called primers, and the primase enzyme synthesizes these primers. RNA primers start the replication process. They serve as a start site for DNA pull. DNA machine polymerase is a big machine that's regulated, and it recognizes these little primers, and it sees an open stretching of DNA that's not anti-parallel bound, and it sets off a copy. And this occurs during DNA synthesis. DNA polymerase binds to these little RNA primers. I'm going to put a short metal base easier. It uses the free 3' end and adds 5' 3' on millions in a row. Uses nucleotides to synthesize the complementary strand. Always works 5' 3'. DNA pull functions by synthesizing phosphodiester bonds. This is a dehydration. It's called a condensation reaction. You're bringing together a phosphate and sugar, and you're losing a molecule of water. Addition of water or subtraction of water is about 95+% of the chemical reactions we do in our body every day. For example, digestion is all addition of water. The pull goes 5' 3' along the leading strand. I'm hoping to give you a picture that adds clarity. The two strands are not replicated the same way because as the fork opens up, one DNA pole sees a continual opening in heaven. He just runs 5' 3' making as he goes. However, the other one runs the opposite direction, and the strand is opening up behind him. He runs the other direction. So that's called lagging strand. I have pictures I hope will clarify. The great Japanese scientist Okazaki is the first one to propose that this DNA polymerase had to act differently on the two strands as the fork opens up. RNA primers are replaced with nucleotides. Covalent bonds are formed between Okazaki fragments with DNA ligase, which sticks together, links up all these little pieces. So leading strand is dead easy. If the strand's running against you and you're a DNA pull and you run 5' 3', as the replication fork opens up and the strands are denaturing before you, you continually see it opening up in front of you. You're driving down an infinitely long tunnel, but the tunnel keeps opening up in front of you. So DNA pull just runs right down. It can add many nucleotides, generally a number on per second. Okay? So it runs down. The other direction runs the opposite way from where the fork is open. Comprehensive? Somewhat? And here's what Okazaki said. This is the great Okazaki. DNA replication co-discovered with his wife, Seneko. Reiji Okazaki tragically died due to the Hiroshima bombs. So he bombed the Japanese at the close of World War II. He was heavily irradiated. He lost most of his white cells. This is probably radiation damage. Our white cells involved in the immune function are destroyed. So he had a reduced immune system. He was very sickly. With only a short period of time, I was able to make this historic breakthrough during this time. So he said the following: as the fork opens up, it's opening up, and remember DNA helicase, it's unzipping this thing, so presumably it's structured, it's positioned here, and it keeps moving. As it slides down, it opens this fork up more and more and more. DNA pull is synthesizing behind it. On this strand, as the fork opens up, this is the guy riding the bicycle down the wormhole, it continually sees this fork moving away in front of it. So it goes, repeatedly runs down the tunnel as it opens. Got it? That's called the leading strand. But, says Okazaki, in the other direction, DNA pull binds to a little primer. He runs the wrong way. The fork opens up behind him. So as he runs this way, a gap appears. Another DNA pull must find and now run up until he butts his head in the back end of this piece. Okazaki says, therefore, when DNA synthesis is complete, One strand will be continuous, and there'll be a finite period of time during which the other strand is short little pieces. And he theorized, based on the size and shape of the DNA pole, which was known at that time, that these pieces would be a few hundred nucleotides long. These are called Okazaki fragments, and they were proven to be correct by experiment. Now, every one of your cells does this. Half of the DNA never stops, and it's made in one long piece. The other half is made in short fragments because the fork opens up behind these DNA poles.
Would that make the Okazaki fragments more prone to like mutations?
Good thought. Very quickly after they're made, they're ligated. It's within a period of minutes. So you can actually isolate these little Okazaki fragments, but after a period of time, an enzyme called DNA ligase is hovering, looking for these nicks. And the primer is removed, it's filled in, and then it's linked together. So the fragments are there for a short period of time. And if a gap occurred, like if somehow a fragment floated away, DNA pole would realize as it moves its proofreading, it's a massive complex. So it's much bigger than this, it's huge. It sees that there's a mistake. Also, a mistake has occurred where a G, say, is paired up with a T. This is an unnatural pair. There'll be a pucker or a mistake in the smooth helical structure. It will recognize it and fix it. This is called proofreading. So generally, mutations are minimal and gaps are ligated together. So it's a good thought, but it turns out, no, that's not the source of most mutation in humans. Actually, remember, only 1 in 3 billion. So every cell will maybe have one mistake in it. That is unreal. I went through all this. This is a small picture showing DNA polymerase. This guy's charging down. This guy is making little short pieces. Here's primase making a little piece of RNA. And the Okazaki fragment runs its head into the back of the next one and then falls off. And actually does this with a series of loops. So it's a single dimeric enzyme, and it forms loops that continually expand and contract during the synthetic procedure. DNA ligase, as a student just asked me, recognizes these gaps and closes them fairly quickly. But long enough so that you can see that they're transient, that they aren't present. Genome, all DNA in an organism cell. DNA contains instructions for the life of all its genes. Humans, some number around 20, 22K exchanges, scattered among 3 billion base pairs. Only about 1% of this, 1.5% is coding. So only 1.5% codes for proteins. The rest does, we're starting to learn, it's micro silencing. There are all kinds of different RNAs which are being made from the rest of DNA. So a lot of it has function. But this is taking decades to work this out. For a long time, we didn't know what all this stuff was doing. Genomics, study of human genome. Human Genome Project, completed in 2003, complete human DNA sequence. And that launched bioinformatics. Transcription. Genes are copied from DNA into RNA. This is the theory of molecular bio. DNA to RNA to protein. DNA is not capable of exiting the nucleus. There's large nuclear pores, but RNA is. So DNA is made into RNA. This process is called transcription. It's catalyzed by an enzyme called RNA polymerase. And all RNA polymerase does is dehydration reaction, and it links many, many different nucleotides into a long string of mRNA. This exits, it encounters ribosomes, massive complex, which makes proteins following the genetic code, which we'll talk about shortly. So this chapter goes through a rip-snorting, fast rip-through of transcription and translation. How do these processes work? All of this is catalyzed by molecular machines. These are protein, occasionally RNA protein complexes that catalyze reactions. Just like little machines, they're sitting there doing something. Okay. Through production of RNA and protein synthesis, DNA contains all the controlled properties of cell and its traits and specific characteristics or properties of any given organism. Transcription, this is taking DNA and making it into RNA. We probably won't finish this chapter, it doesn't matter. And I apologize, these two chapters are particularly tough. It's assumed to be somewhat a review, so if you're completely, totally, thoroughly lost, spend some time studying, or ask me, or ask friends. This is the central dogma of molecular biology. This process is called transcription. This is heavily controlled or regulated. This process. Only some DNA is transcribed. Remember, you have about 22,000 genes, and each cell at any time is only expressing about 5,000, so about a quarter of them. So some process is saying a muscle cell is going to make these genes, and a nerve cell will make these, and a bone cell will make these. They're not going to be the same. Slightly different health. This is all regulated. Each cell is not expressing the same set. This is called translation. This is RNA polymerase. This is called the ribosome. This is going to be lightly fast. Transcription, RNA pol binds to the promoter. This is a sequence in the DNA. Unwind, separate strands, template strand is copied by RNA polymerase. It's usually called RNA pol, DNA pol. Copies template DNA 5', 3'. All synthesis on this planet, whether it's RNA or DNA, is always made from the 5' end to the 3' end. 0 exceptions. Uses nucleotides. Adenine uracil now appeared. Remember, the methyl group's gone. I'm finding why, I don't know. But RNA uses a unique uracil, U. U pairs with A, just like the T did in DNA. A pairs with U and C with G. This means there's 2 hydrogen bonds, and C, G actually has 3 hydrogen. Slightly strong. Ribonucleotides are connected by phosphodiester bonds, and now at the 2' position there's a hydroxyl. DNA is deoxyl. The end of a gene RNA pol, Cannon's termination sequence. Loops are created. This is part of secondary structure. You have anti-parallel hairpin structures within RNA. RNA is actually fairly highly structured. Newly formed strands of RNA are released. RNA strand is called messenger RNA or mRNA. It has to mature in eukaryotic cells and our cells, matures into a form which is capable of exiting the nucleus and being translated, making protein. Proteins are life. Proteins are the enzymes that give your cells metabolism and energy and life. Multiple copies of RNA are transcribed from each gene, and they're zipped off large numbers at a time. And this actually is conditions. So this is showing some gene. Here's RNA pol, it binds, the gene starts to open up, just like it does at an elongation fork. This is called initiation. It's binding to what's called a promoter. Promoter or sequences that say, RNA pol, come and bind here. They're going to transcribe this gene. Each cell is doing roughly 5,000 genes. This thing elongates, and as it goes, the double helix closes behind it, and off zips a growing RNA chain. Note, does RNA pol make Okazaki fragments? Why not? Lauren?
It's not splitting into 2 strands. It's not splitting into 2 strands.
Sort of. There's a simpler answer. I would argue yes, because you're still opening this up. This is just like the replication fork of DNA. Your DNA helix is still opening up. The anti-parallel sheet is coming apart. You're creating a gap. RNA pole sits there, zips forward. Does it make Okazaki fragments? The answer is no. Why? Anybody else? RNA, it wasn't implicit in the answer you gave. RNA only catalyzes one strand, which is called the sense strand. The other strand is not copied. So all RNA pole always runs down the worm tunnel. It's always seeing the fork open up in front of it. Only one side is made, not the other. The other, that would be like, if you're reading from left to right in the page, if you read in the other direction, it doesn't make any sense. Unless it's a palindrome. The answer is no. Okazaki fragments only occur when you're opening up a fork and replicating both strands, forward and back. RNA is not the only type. Ribosomal RNA and trans-RNA are also made. Do not carry information directly post-synthesis. Ribosomal RNA is structural component of ribosomes. Trans-RNA interacts with DNA template. This is part of the genetic code which comes out. New class of microRNAs, and there's many new classes are being discovered almost every year. RNA processor. I know, please stay focused, try and follow it. So RNA in the eukaryotic cell, once you've made it and it's getting ready to exit, it has to be modified. So you have all kinds of controls that mature RNA until it's a mature messenger RNA that can exit the nucleus. These modifications occur before primary transcripts exit and are ready for ribosomes to synthesize. Number one, splicing. This is what I said before. Most genes in us, that's what racing is almost done. Most genes, if you use a long sequence of DNA, here's the promoter, here's the start site, start. Most genes occur in blocks. There's a block, another block, and then maybe a block over here, and a block down there, etc. Calcium channel, I think, has 41 blocks. These blocks are called exons. The intervening sequence, this is an exon. The intervening sequence is called an intron. A system, biggest molecular machine in our cells, is the spliceosome. And it goes through this, and it links exons together, and then you're skipped. So if there's a couple in here, they skip these, and go to here. So the 41 blocks are not made the same in every cell. This means that one gene can make multiple proteins, which are sequence distinct. Calcium channel in a muscle cell, and in a immune cell, and in a neuron will not be the same. They'll look different. Okay? That's called splicing. Let's pick up here next session. Quiz number 2. We have a quiz probably at 9 o'clock next week. And the quiz will be everything up to this slide. And it covers all what we've gone over today. I don't think there's much else. We're going to reconvene here in 30 minutes for lab at 12:15. It's a relatively quick lab unless you really screw it up. So it's up to you guys to determine how long the lab is. If you are successful, it will be about an hour. If you're unsuccessful, it could be midnight. Good luck. right here this year.